Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. Today's computational neuroscientists are lucky to live in a time where important new experimental data becomes available. New experimental data is almost a guarantee for assuring that the scientific field is vibrant. In contrast, I think today's theoretical particle physicists are envious of their past colleagues who worked in the 1950s and 60s when new experimental data regularly came along. One example of new neuroscience data is the large-scale recordings of electrophysiological data with new electrodes such as a neuropixel probe and optophysiological data from tens of thousands of neurons found using 2-photon calcium imaging. In podcast number 24, Andreas Tolias talked about how his group has taken advantage of optophysiological data from 135,000 neurons to make so-called foundation models with unprecedented ability to predict responses in new situations. But also in neuroanatomy, giant steps forward have been made. A few months ago, the so-called connectome of a whole fruit fly, mapped out using electron microscopy, was published. This includes nearly 140,000 neurons and more than 50 million synapses. And in the so-called Microns project, the anatomical structure of a cubic millimeter of mouse cortex has been studied in similar detail. It will be exciting to see what new insights will come out from analyzing these new rich sources of information. More than 100 years ago, Santiago Ramón y Cajal pioneered the experimental study of neuronal morphologies and revealed the complex dendritic structures of neurons. This is often said to be the start of modern neuroscience. In the 1950s and 60s, Wilfred Rahl explored the functional implications of dendrites using the cable equation, initiating what is now called biophysically detailed or multi-compartmental modeling of neurons. In podcast number 15, Michael Hines talked about the simulation software Neuron, the most commonly used tool when doing such modeling. And in podcast number 17, Jota Poirazzi talked about what we have learned about the role of dendrites in signal processing by doing such simulations. To do biophysically detailed neuron modeling, a detailed dendritic morphology is needed as a starting point. They can be obtained by anatomical reconstruction of real neurons using light microscopy, and now in with electron microscopy. This is a tedious process, but fortunately there are now databases where such reconstructed morphologies can be found. An example is the database neuromorpho.org, which contains morphologies of more than 270,000 cells. An alternative is to grow your own dendritic morphologies using simulation tools, and this is the topic of today's podcast with Hermann Cuntz. He has been working on this for several decades, and in 2010, he published a key paper with a cool title, One Rule to Grow Them All, a general theory of neuronal branching and its practical application. Here he not only published his new growth algorithm and some examples, but also an open source software toolbox called Trees for generating various types of dendritic structures. In the podcast, Hermann, now leading a lab in Frankfurt, discusses the many benefits of being able to virtually grow dendrites, and also describes the inner workings of his growth algorithm using principles that go all the way back to ideas from Ramón y Cajal. In later work, Hermann's group presented another software tool called T2N for interfacing Neuron and the Trees toolbox. This tool allows for generation of biophysically detailed neuron models, stable over a broad range of reconstructed and synthetic morphologies. We also discuss this work and its application to generate active models of dentate granule cells, replicating a wide palette of experiments from various labs. Finally, we discuss some work that Hermann and collaborators have done on the overall principles of why dendrites look the way they do. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking. For example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you also can watch the videos. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Hermann. So, welcome to the Theoretical Neuroscience podcast, Hermann Cuntz.
Good morning, Gaute.
So, I saw from your lab's webpage that you are actually leading a group in computational neuroanatomy in Frankfurt, something called the Ernst Stringmann Institute, and in cooperation with Max Planck and Frankfurt Institute of Advanced Studies. Actually, that's something I've seen with German scientists. They often have a lot of affiliations. I don't know, it has something to do with the funding. Maybe it's just.
That's correct. No, I'm actually a lecturer in Gießen University, so I have even one more
Yeah,
affiliation. Now
already.
I'm speaking indeed from the Ernst Stringmann Institute, which is a privately funded Max Planck associated institute for neuroscience,
Mm.
indeed in
Yeah.
Frankfurt. And then we have very strong links to the Frankfurt Institute for Advanced Studies, where there's a lot of the theoretical neuroscience community, and further links to the neuroscience computational neuroscience network in
Yeah. So
Germany.
you seem to have a quite strong environment in Frankfurt in computational neuroscience or in neuroscience in
Indeed,
general.
indeed, and also historically, Frankfurt is very strong with the Max Planck Institute for brain research in
Mm.
neuroscience. So that's
Mm.
very
And then
good.
Gießen is good because our friend Peter Jedlicka
Indeed,
is,
yes. So
yeah, so
Peter
that's not so far
Jedlička
away.
and me, we share a lab, and so he's professor in Gießen, and so I'm affiliated with him there,
Hmm.
and we spend a lot of time also in Frankfurt, so it's
Yeah.
nice.
Mm. Good. So your group is in computational neuroanatomy. So what is computational neuroanatomy?
So in a way, it's kind of a almost a forgotten subject because computational neuroscience, very broad field, right? That covers basically all of neuroscience, and there's computer models and theoretical models for basically everything from cognitive processes in the brain all the way to molecular processes and of course cellular stuff that we are interested in. But interestingly enough, many of the modeling studies are being done at the electrophysiology, at the functional level of the cell, but the anatomy matters a lot as well, and so and has not been considered as much as it probably should.
Mm. Yeah, but I guess it's now coming back a little bit. I mean, there's all this. Just a few weeks ago, there was all this. There's all this fruit fly connectome, right? And there was
Exactly.
all this like
Yes. So
series of
it has been
papers.
already coming back before the connectomics trend because a lot of new microscopy methods are available and genetic tools to target individual cells and so follow them. And it has become really relatively easy to to image cells also, through development and stuff. But now the newest technology is these serial electron microscopy methods where at nanometer resolutions, an entire circuit can be mapped with all of the morphologies of all the cells, but also the synapses, so the connections between the cells, everything. And now we have data for the whole fly brain, fly larva, and also larger chunks of mouse brain, etc. Yes, and so indeed there has been just recently one whole Nature edition with I think nine papers on the connectome, and a lot of cool stuff coming out. Yeah,
Yeah.
yeah. Also
So that's
relating actually these anatomical the connectivity with actual electrophysiology and electrical activity.
yeah. I guess that's just whenever there's like important new data coming in, that's always a good sign for the field. That means that it's a vibrant thing, new things are happening, and also for like modelers like us, we actually get sort of more data to work on. So
Exactly,
that's good.
and there's this very nice new trend that um, of open data, right? That people are now really sharing the data, also the models, of course, but also the data, and so we can just download some of the data. That
Yeah.
doesn't mean that collaborations with very good experimentalists aren't important, but we can already as these big connectome data sets, you can just download them or scroll through them on the internet.
Yeah,
And
and I think it's just it's a wonderful sort of in some sense like a democratization of computational neuroscience that you can sit anywhere. You don't have to be associated with a big lab.
exactly.
It's kind of you need sort of computers and you need your brain and your a fire in the gut, whatever interest. Then you can sit
Yes, and there's also
anywhere.
tools for modeling that are available for everyone, like Neuron and Brian and Ness, all kinds of tools to actually
Yeah,
study the electrophysiology. Right?
yeah, and then of course also have to mention this eBrains
E-brains,
initiatives
indeed.
or like for
Yes.
to make this makes this feasible. So, but your focus has been on dendrites, and dendrites, of course, is maybe has the proudest history in of all in neuroscience because it goes back to Ramon y Cajal, the sort of like the father of modern neuroscience.
That's true. So you were actually mentioning that right now we're in a period of extensive new data on the with all these new techniques, and I think that Santiago Ramon y Cajal, he lived in a period where basically all these new staining techniques were developed. And people spent a lot of time looking at slices of brain that were stained with different techniques, right? And he was one of those who actually sat then in their kitchen, looking at slice after slice and trying to figure out what you can see, what you can infer
Mm.
about the brain by looking at those.
Yeah, and I guess also, I mean, he obviously I mean, he was very he was drawing all these beautiful structures, right, based on
Exactly.
what he saw in the microscope.
So
And
he was a painter. He loved painting. I'm
yeah,
not sure, you can judge yourself uh, if you like his original, paintings of still lives and stuff. But the drawings that he does of neurons, they are so beautiful and they're
yeah,
hanging everywhere.
yeah,
If you can
exactly.
recognize them, you'll find them everywhere.
That's true because they are like on so many front pages of neuroscience books, right?
That's
Yeah,
correct.
because for
That's
good reasons, and they're
correct.
really beautiful. And of course, they also discovered this that they are really all different. It's like a really rich world of dendrites, and I think they call them. He or someone else called them the butterflies of the soul. Very poetic and beautiful, right? This is
That's
sort
right.
of it's
Yes.
sort of the most beautiful entity I think in neuroscience, right? This some of these dendritic
Yes, and
structures.
actually, for us, it has been quite nice as well because it helps, to get your work into onto the cover of journals or even into museums,
You know,
even famous
actually,
ones.
that's true. I was talking to Torbjörn in our group yesterday, and when I said I was going to talk to you, and so Herman, he has made all this beautiful neuron art that has inspired him also, like for
Excellent.
yeah. So for
Excellent.
when we sort of make sort of like so
Yeah.
that's true. So
Yeah.
that's sort of like the poster girl or poster boy of neuroscience. That's good.
Yes, the dendrites are
So,
indeed
so I
beautiful.
mean, then of course Ramon, well, Santiago Ramon y Cajal. It's sort of a coolest name in neuroscience, also. So, but he sort of discovered all this beautiful anatomical structure of the dendrites, and then I guess Wilfred Roll was the one who sort of took it and sort of demonstrated that it also has an important the branching of the dendrites is important also for to understand the function.
Yeah. So interestingly, Cahal already, just by looking at the morphologies, was able to infer some of the function, and so he was able to identify, the dendrites as the input structure for the cells and the axons for as the output structure, and so therefore he was able to look at circuits and draw little arrows because input output, right? And so infer, for example, sensory processing, what is the cascade, and hippocampus learning, what are the stages of propagation of activity, and but then indeed, I think when electrophysiology came up, right, then you had these recordings from the soma of the cell, and they were regarded, I guess, the dendrites were rather disregarded as basically just something that collects input somewhere, right? And
and also I think part of the like the doing the other metabolic needs, serving for some other non-signal processing reason.
Exactly. Which certainly they do, but so they were ignored, and then Wilfried Roll was really the one who showed that they are important. That if you consider current transfer between the synapse that's far on a dendrite and the soma where all the signals get added up, that the specific structure and where the inputs come from, that actually matters. And now electrophysiologically. Yes.
And based on then both the work of Wilfred Roll on dendrites, and obviously also the work of Hodgkin and Huxley on making these models for the active conductances, we could then start doing multi-compartmental modeling of like biophysically detailed neuron models. And for that, you need sort of obviously dendritic morphologies.
Yeah.
So, I mean, one thing, now we talk about connectome, but some sense, you've been particularly interested in dendritic morphologies, and this is sort of how I got to know your work. So maybe have you introduced the term dendrome for all kind of possible
dendrome?
That's sort of for all kinds of possible dendrites. Is that it was a time when you had like proteome and genome and connectome and all this.
No, it's an interesting idea, but if we think about something that covers this general space of dendrite morphology, we talk about the morphological space, for example, of all possible dendrites of a particular type. So if you take, for example, a pyramidal cell, there's a restricted kinds of morphology that the pyramidal cell can take over, and the space of all the possible morphologies that you would call the morphological space of those.
So I think the starting when I did I your paper from 2010 on sort of history how it had this cool name, one rule to build them all or something.
One rule to grow them all. Yes.
One rule to so that's
yes. It's reversing kind of the Lord of the Rings. The rule
that's
is
good. Yeah. Okay. Exactly. Yeah. Exactly. Very clever. No, I remember. Almost at the rate I remember sort of the.
I was hoping they would take it as a title, and they did.
Yeah.
They did not complain.
That was cool. Anyway, so that you sort of actually had this. You introduced it was the paper was on about how to virtually, essentially, in computer simulation on models, grow dendritic structures, and you also introduced this, I guess, this trees toolbox, the MATLAB toolbox. So the first question is sort of why do you want to grow dendritic structures?
Why model, right?
Yeah,
Why model?
why model
Yeah.
dendritic structure? Yeah.
Obviously, I mean, when you model anything, you'll understand it better, right?
Mm.
With dendrites, we find that there's tons of things that we can do using the morphological models that we have. So what we have is basically models that describe the shape of a particular dendrite type, right? So different neurons have different shapes of dendrites, and so then we have models that describe the different shapes with small numbers of parameters, so that we understand the models also very well, and the rules that the dendrites use to grow, we understand them relatively well. So things we can do that maybe sounds a little bit boring, but we can of course use the model to understand statistics of branching, so all the descriptions of and their scaling relationships. So for example, if you have a longer dendrite, it will branch more, right? And we have very strong predictions, and this is also something that we see also in the data, so that we can understand, right, this morphological space, restrict what is possible. Not all possible trees
Mm.
are dendrites. And so having a model of a particular dendrite type, you can study all its relationships. We can also use the models to study actual growth, so during development or maturation in the adult, or even changes because of lesion or something like that. Um, we can um, we can generate larger networks built of realistic morphology, so models that can, so these compartmental models that you mentioned earlier,
Mm.
and I think you had a separate podcast on this. Uh, they they of course depend very strongly on the morphology, and if you have only 20 drawings, reconstructions of dendrites, then you can make only a small model, or you copy paste the morphology if you have an electrophysiological model, right? But if you have a morphological model, then you can actually generate millions of new morphologies, different morphologies, and you can also strongly relate um, the connectivity and the morphology. This is also something that our models actually help to do. So we don't only copy paste the morphology, or generate morphology in empty space. We can generate morphologies with respect to the connectivity that they actually implement, which then allows us again to link our models to these connectome data, right? That we talked about earlier. And and of course, the most important thing that we can do with our models is to combine them with compartmental models, electrophysiology models, and then study the structure-function relationship in
Yeah.
detail, right? So we can
So,
say change parameters in the morphology and see what difference it makes for the computation that the dendrite performs.
because that's sort of the application I know mainly, right? I mean, if you want to make a multi-compartmental or like a biophysically detailed model, you need the dendritic structure, and you either have to get it from some kind of experiment, sort of like reconstructions with neurolucida or something. And at
Yeah,
least back in the days, there were very few of those neurons, right, or the
that's true.
reconstructed neurons. Now there are much more of them in databases like Neuromorpho, for example, and also other databases. Or you must grow your own structure, and that actually also that's how we collaborated on a paper once, right? When we
Yes,
sort of we want
that's
to
right.
study how the local field potential generated by dendrites depend on the dendritic structure. We want to sort of like to explore that, and then it was very practical to have this to grow the structure so that we could sort of essentially keep everything the same,
Yeah.
except in that case the length of the apical dendrite.
And I found it actually, that particular project very interesting because um, using the morphological model, we saw that you can change just one parameter and roughly change a stellate cell into a layer 2-3 pyramidal cell in cortex, layer 4 cortical pyramidal cell, or even layer 5 cortical pyramidal cell. And it's basically one parameter to change.
Yeah.
Interesting.
Exactly.
Yeah.
It leads to a better understanding, and also, it sort of reduces the complexity and something in our understanding, that it's actually you can understand these things by, in that case, a single parameter.
Yes.
Okay, but so and I guess also one application is maybe to, if you is it also to, if you have some kind of partial, I mean, you have done some anatomical reconstructions, but you miss something. Maybe you can sort of, can you do this, sort of grow the missing part or something?
Actually, so we have just one of our most recent papers is about repair, and okay, so you can of course use the algorithm to grow missing bits, but one thing that and we have the tools for that. But one thing that I found very interesting here is, so we haven't talked yet at all about how the model works, but
No, we're coming to that.
let's assume we have a growth rule. The growth rule that we propose actually explained the regeneration of dendrites that were cut experimentally with a laser, right? So there is experiments in fly neurons from the Jan and Jan empire,
Okay.
where they fly larva neurons where there's a laser ablation of subtree, and then there's regrowth, and our model predicts that that in some cases there's regrowth from the cut branch, and in other cases there's regrowth from neighboring branches. And this bimodal distribution is something that was actually proposed experimentally, and we see it in the model.
Because you're now approaching coming to your model, because it's your model is, at least the one in the trees toolbox, is statistical in the sense that it doesn't model the mechanistic things with growth cones and tubuli and like these molecules. Right. And these things. This is the goal is, some sense, to use then statistical information from the observation of neurons, dendritic structures, and then sort of to grow like similar kind of like statistically similar kind of neurons, or
so that's what other people have done. So there's some really nice work, previous work from Jaap van Pelt, for example, and Giorgio Ascoli that have generated very nice morphological models based on statistic. So growth, and then fit the parameters of a statistical growth model so that you get morphologies that look like
so these statistical, so that's like shape parameters of like neurites, like how the length and diameter and when they branch and stuff, like
how they branch, and there's self-avoidance that plays a role, and these kind of things that you can put in, and then you have that every new constraint that you put in gives you one more parameter, and then you'll have 20, 30 parameter models, and then you can find the best parameter combination. Right?
Yeah, and then so what you're saying is that this is the we can call it like the traditional approach. At least it was before 2010.
So traditional, yes. It's hard to call it traditional because we're such a small field, right? But indeed, yes, this was kind of a inspiration for me as well. Like I saw these beautiful work from Giorgio Ascoli, these fake neurons that looked absolutely amazing, and were inspired by actually Lindenmeyer systems. So it's a kind of a stochastic version of a fractal growth, and I really wanted to be part of this. But then the model that we designed, I think it has the nice advantage of following really very simple rules, while it still we completely understand, while it still predicts the full details of the morphology.
So it's simpler.
So our models
conceptually simpler.
Yeah, they're based on the idea that optimal wiring determines basically the morphology of
Yeah.
dendrites. And this had been proposed already in the past, particularly by Mitya Shklovsky, who
Mm.
showed that the math and basically statistics, so scaling relationships and such are consistent with dendrites and axons. Um, Conserving material, basically, being optimal in terms of the wiring,
Mm.
but they were really mathematical models that just gave equations, whereas we then use this idea in principle to generate fake dendrites,
Mm.
real fake dendrites
Yeah.
that you can study then.
Because I think I read in your paper that also this Ramon y Cajal had some of these ideas for laws of dendritic architecture. There was like some
That's true. That's
optimization
true.
principles.
So originally, so Ramon y Cajal, he left us with with two huge books that are wonderful and actually really I really recommend to look into those.
Mm.
um, and they are the result of him drawing again and in his kitchen,
Mm.
all kinds of different dendrites and axons from different developmental stages in different species, etc. And in the very beginning of his book, he says, well, I could not have done this
Mm.
if I hadn't realized early on that actually neural circuits follow some fundamental branching laws, and they are relatively, I mean, intuitive, if you want.
Mm.
So the assumption is that you have conservation of material, so you
Mm.
won't use too much cable to
Yeah.
reach a particular location.
Because it takes resources to just like the size of the cell
Right, keep the
keep.
cells small while it still has to it still has to extend. So the
Sure.
dendrites are these very nice
Mm.
tree-like structures. They
Yeah.
have to
Mm.
go to collect the inputs from other cells, so they have to span a particular innovation area, but
Mm.
they need to do this efficiently. So
Mm.
they actually use little amount of basically cable to do this.
Mm.
And the other thing is for neural systems, of course, conduction time matters a lot, right? And so
Mm.
that the path between, let's say, the synapse and the summation point of the signals, that should be relatively short as well. So if you go in a long spiral before you reach the soma, that's
Exactly.
not efficient, and so
Mm.
that also matters. And then the third rule that he came up with relates to basically packing of all these branch structures in the brain, so that the brain in total isn't too big, because it's already
Yeah.
quite big as it is.
Yeah. Exactly. So exactly. So you have so you have principles for how to make a. So in some sense, each dendrite gets an allotted genetic structure, gets an allotted space, constrained by the size of the head, maybe, and the number of neurons you need. And then within that space, how does the individual neuron optimize the sort of making a dendritic tree? And you shouldn't make too. You shouldn't sort of be wasteful and make branches without synapses, for
Mhm.
example. That's just a
That
waste.
would be big ways. Yes,
Yeah,
those we
exactly.
could not explain with our model
Exactly.
either. So
So that's sort of exactly that. So you never see around the big city that there are big roads going out to nowhere. Actually, some places in the Gulf, I think it is like that because they're waiting for development. But generally,
That's
most.
true. So in a way, actually, cities are good, and street networks are a good
Mm,
analogy,
uh,
because actually, graph theory that applies to these kind of networks
mm,
is exactly also what we then use to find
mm,
the best possible um, dendrite structure to perform these circuit requirements.
yeah. And okay, so now into your algorithm now, because one important concept here is this what they call carrier points.
Mhm.
So what are carrier points in your algorithm?
So, so I mean, they're kind of originally they're kind of a fake thing that was useful. So we just assume, okay, again, we have this innovation area, and there will be some locations of the inputs that the dendrite has to collect, right? So it has to go there, and then you can then, if you have those carrier points, then you can try to find the best graph that connects those to minimize, for example, the total cable, right? Um, and so first we were very careful not to say, they're not necessarily the synapses or something, right? But it turns out in some of the work that we did afterwards that at least statistically, the number of carrier points that we need to generate a dendrite actually matches quite nicely. In some cases, also the number and distribution of actual synapses, and we can, for example, also use synapses from the connectome data set, and then generate fake dendrites based on the synapses as carrier points. So you have to be a bit careful not to make the assumption, yes, they are the synapses, but they relate somehow to the synapses.
So that's the carrier point. So you first start up, okay, you have defined this like area, just for simplicity, it's like say it's like a circle, 2D circle,
Right.
and then you fill this 2D circle with carrier points, and you can maybe think of them as sort of positions where synapses connect, but that's sort of like not completely true, but like a good way to think about it, maybe. And then, but then you sort of to put these carrier points out there, then you need some idea about where to put the carrier point, right? I mean, it's like I think you introduced something called the
Right.
density profile.
Yeah, so there's then all kinds of different ways that you can do this. For example, you can take an actual data set of real dendrites, or if you know where the synapses are, the actual synapses, and then scale them so that they all fit into the same context, and then calculate the density profile, and then use the density profile to distribute these carrier points then randomly inside. And then use the carrier points to connect. So and the connection actually, so this is important. They are then connected using kind of a version of the minimum spanning tree algorithm, which is an algorithm from graph theory. A pure minimum spanning tree actually will give you the edges that connect all those carrier points that in the end uses the shortest total cable length. And that is one of the costs. One of the costs also from Kahal, right? That's the conservation of material, basically the first rule. And that is not enough to describe the dendrites. So you have to add one more cost, and that is the signal conduction time. So we use the path length. We minimize the path length within the same algorithm, and so then we have basically one parameter that says how important is the path length or how important is the material, and that parameter we call the balancing factor between those two costs. And then if you choose a balancing factor adequately for a particular cell type and distribute these carrier points, we also sometimes call them target points, adequately, then you can get very realistic looking dendritic trees that also follow the correct statistics.
Yeah, because I guess if you think of that, then you have this important balancing factor. So say it was balanced that all that mattered was sort of the path to this, say, soma, right?
Then every carrier point will be
Yeah.
connected
So it would
straight
be
to
like
the root.
this star or whatever you
Right.
call it. It's like
Which is unrealistic, although some cells have a very high balancing factor, like
yeah,
granule
yeah.
cells.
So
They send,
so
and then you can see that already.
yeah,
So this is a very visual thing. We're trying to
yeah,
explain it
yeah,
in words, but I
yeah,
think you can imagine that if you have these direct connections to the root, that basically that means the dendrites, they all grow away, straight
yeah,
away from the
exactly.
root. So it's you can see
Mm,
already
yeah,
visually
yeah,
what
of
will
course.
be a good balancing factor for
Yeah,
different cell types. And different cell types do actually have different balancing factors, which also means that functionally that is actually short conduction times are more important for some cell types and less important for others.
yeah. Hmm. So if okay, so what about the other extreme? If only if yeah, exactly. That's if only the but if only the material matters, the amount of material, then that would be then it could be really slow to get from some points to the
Yeah,
soma.
then you get these spirals around,
Yeah,
which
yeah,
then a signal, it would take forever to reach
yeah, yeah.
the soma, and neither extreme, like the
No.
low balance of factor, nor
Hmm.
very high, actually really exists. So it's always something in between.
That's true. But I guess if you think of it as a city architect, right, who wants to make sure that everybody works in the one building in the center city of the center, how
Right.
do you put out the roads so that people don't spend too much time, but not that everything well not each house can have their own
Yeah.
private road to the center. So you have to find a compromise, just like the
Right.
neurons.
And dendrites are obviously they are trees, as you'd already allude to. It is one point that's important, right? The soma, and
Mm,
so there are no loops, as opposed to the street structure in a city, right? That makes them relatively easy also to study and to model. And another nice character of dendrites is that they basically only ever have bifurcations when they have branch points. So there's
mm.
never four branches coming out from the same branch point. It's
Okay, no,
um,
that's I guess that's true.
sometimes very close. Two branch points are very close, and you'll
Yeah,
have what looks like three maybe branches,
yeah,
that's already quite rare. But so it's basically bifurcations only.
mm, yeah. Okay, so when you start growing this, then you have this. You have to start somewhere, right? Which will be your soma,
Right.
right?
So the minimum spanning tree algorithm
Mm,
works in a very nice way. There's a greedy way that says you always connect the target point or carrier point that is the closest to the tree
mm,
to the node on the tree to which it is closest to.
mm,
And if you do this iteratively, you grow your tree from inside out,
mm,
then you will get the minimum spanning tree. This is true for the cable length
mm,
minimization. We use the same algorithm, so iteratively from the inside out, and add this conduction time or path length cost, right?
mm, mm,
There, then you cannot prove mathematically anymore that this is an absolute global optimum. They're still very good, but they're
mm.
sometimes not the absolute globally the best. That's not the way to get this
Yeah, I mean, you mean this greedy algorithm is not
right, the
necessarily
greedy
the best.
algorithm.
Mm,
But it's still very good, and so basically that algorithm grows from inside out. Now, development actually often doesn't do that, right?
mhm,
And so we've had a very fruitful collaboration with the lab of Gaia Tavozanes, who looked at dendrites in the fly larvae and how they grow, so um, in many cases, the dendrites will if there's a spanning area, there will be first branches that kind of go as far as possible to
okay.
cover the area, and then there will be filling in
Ah,
branches iteratively. And so we actually managed to describe also this model because of the very nice data. And that's nice because now we can actually say, okay, we think we understand how biology implements this minimum spanning tree algorithm
Mm.
problem, right? And it's probably better than our greedy, not completely perfect algorithm.
Uh,
And
that's good.
so it's an elegant solution. And in the end, you can also show that during the development, actually, um, also the in between stages are still optimal with respect to the
Oh,
cable
oh,
length, right? So that's actually
it's a
quite a nice thing.
clever rule.
Yeah,
Yeah.
clever biology.
Yeah, clever biology, exactly. Clever biology. Clever rule made by biology.
Yeah.
Yeah. So okay, but then I mean, some in practice when you use this rule, right? Because if you look at these dendritic structures, like in some cells have I mean, there's somas in the middle of a 3D structure, maybe like these stellate cells, or
Right.
and then you have some, well, you have some cells where you have like this big soma, and like and in one end of the in one yeah, at one edge of the dendritic structure, and they have like maybe this is not like these Purkinje cells often have like
Right.
this nice fan of beautiful dendrites, but it comes out the soma is not in the center. So so
Right.
this is somehow had to go into your growth algorithm.
Yes, exactly. So the geometry of it, you have to kind of describe in such terms, and how you do this when you do the modeling is a little bit up to you. um, uh, the Purkinje cells, it's very nice because they have these the very large number of inputs, and they come from granule cell parallel fibers, right? There's the axons from the granule cells in the cerebellum that are all really parallel, completely parallel to each other.
Mm.
And so then if you grow a morphology to collect inputs, let's say, from all of those parallel fibers, then with our algorithm, then automatically you will get a two-dimensional dendrite that's perpendicular to the parallel fibers,
Huh.
and that's also exactly how Purkinje cells look like.
Hmm. Huh.
So they have they are flat and they lie exactly perpendicular
Sorry,
to
why
the
so what assures the two dimensionality? Why doesn't it
So, so the it is obviously you need more cable,
Mm,
more material if you would be slanted, right?
yeah.
So if you're perpendicular, you use the least amount of cable. It's this again a little bit a visual thing. Maybe if you need to look maybe
I have to look at the video. We have a beautiful video of
but
the
the other thing is,
Hermann's
is
hand
of course
here.
conduction time. It would also be minimal if you actually go perpendicular. So
Hmm.
both costs actually force the cell to be perpendicular and flat.
Huh.
And in other arrangements, like for example, then granule cells in a dentate gyrus, the input axons are a little bit less than perfectly parallel, right? And then the granule cell, instead of being completely flat, it's kind of ellipsoid or maybe even actually cone-like, and so depending on the input organization, how the
Mm.
axons come in, the
Yeah.
dendrite will also grow in a different way. And I think
Oh
that's
yeah.
strongly determines the differences in dendrites for different cell types.
So exactly the input structure, the inputs gives this essentially where the synapses are, and that gives you this what is called this field, you call it, right, the density field, and so it tells you the carrier points will in sort of be so I guess if you think of if I think of like our favorite neuron, the pyramidal neuron, uh,
Yeah.
yeah, that's sort of probably the most studied neuron in terms, at least in the modeling world. So pyramidal neurons in the cortex or hippocampus, it has sort of like these basal dendrites and its long apical stalk, then maybe, and some apical dendrites at the top. Then in order to grow that, you sort of take a bunch of or how do you grow? So you use
So that's actually an interesting cell type because
mm,
um, Actually, the pyramidal cell gets inputs from different layers, right? And so maybe simplify it for two layers. If you have input axons and two layers, and you want to get you want to send your dendrites out, then
You mean like they have received synapses? They have receiving synapses in two layers, like an
yeah,
apical and a basal, or something.
right, and the soma is in one of the layers, right? And the soma location in our hands using our model determines basically where the basal dendrite is, and the other layer will contain the apical dendrite. Now, why? So all the pyramidal cells, they all have these basal and apical dendrites, and they're all polarized in the very same way. Why is that? That's again probably because of optimal wiring, because the output, the bigger axon output of the cell goes to then the white matter, which is always on one side of the pyramidal cell, and so that therefore all the basal dendrites are on the side of the white matter, and all the apical dendrites will be on the other side.
Oh, so the axon from the pyramidal cell itself actually affects its
Right.
own
The
dendritic structure.
location of the soma, right?
Yeah,
So that's probably. I mean,
yeah, exactly.
in the toy model that we developed, right? So
Yeah,
this would be kind of a prediction from the model, let's
yeah.
say.
But I mean, the pyramidal cells also sends axons up, right? Up
That's right.
in,
Yeah,
yeah.
yeah.
So,
So,
but
so, so the
it's
local axons maybe don't matter then as much.
okay.
That would be a prediction from here,
Yeah.
because indeed you can put this soma. So if you put the root for the growth algorithm in the other layer, then what you get is an upside down pyramidal cell. And there are no upside down pyramidal cells, so very, very few in cortex. So where the soma is really matters for how the shape of the
Yeah,
cell
because
is.
the interneurons, they in the cortex, they have they are sometimes. I mean, they then don't have this very, but they have also this often these long dendrites. But this somas are all over the place, aren't they?
Yeah. So we didn't look as much
No, I'm just thinking because
into this.
they
Yeah.
don't, since they're interneurons, inhibitory interneurons, they don't project out,
Yeah,
right?
so,
So
so
maybe
they
they're
exactly, they're more local, and
yeah,
therefore and they have very particular targets.
exactly.
If it's
So
a
all bets are
specific location in the axon
yeah.
of other cells, or a
So
specific location in
all
the
bets are off.
yeah. Exactly. And
So
that's really why Ramon y Cajal was able to to identify the cell types just by looking at the dendrites, because they are shaped so differently, and the shapes are as we're talking about really, like, extensively now, they are determined by basically these circuit requirements,
mm,
very
mm,
strongly by the circuit
yeah.
requirements.
Mm. But what you get out of your algorithm is a graph, right? You don't get out diameters.
No, exactly. So
So
that's a
so
skeleton,
yeah,
basically the nodes and the arrows between the nodes,
yeah,
basically the edges, and so it's
mhm,
a graph. Yes.
yeah.
And
So how do you when you have your when I got the model from you, well, when for our project back in the days, then we got diameters and everything.
yes.
Yeah.
So
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 what did you do there?
already with the very first papers where we did morphological modeling, we had also considered the diameters, which you can basically map separately once you have the skeleton.
Mm,
And at the time, so this was a bit computationally intensive. We looked mostly at cables, and we found that if you uh, so if you want to be sure that distal synapses are heard at the soma as much as proximal
mm,
synapses,
yeah.
um, even though they're far away, and there's the signal actually becomes
Mm,
less
mm,
according to Wilfried Rohl's cable
yeah.
theory, right? Then you can generate a diameter profile along the cable that actually kind of democratizes this, so makes the distal synapses heard as much as possible. And what comes out there is that there is a tapering, which is also exactly what we see in real dendrites. The dendrites are tapered, which means they're thinner towards the tips, and this tapering is a quadratic taper. So there's a quadratic relationship.
So what does that mean? That's
square,
yeah.
basically. Yeah.
So meaning
So
that
parabola,
parabola, so that it gets thinner.
A bit surprising. And
So what do
in
you
the
mean
meantime, we
parabola?
also had so Alex Bird in the lab. He's a mathematician, and he actually calculated that analytically using the cable equation from Wilfrid Rod. So he came up also with this quadratic.
Sorry, what do you mean quadratic? I just want to make sure. So quadratic means that the distal end of the dendrite is it
so it becomes thinner and thinner, and it grows towards, let's say, the soma in a quadratic way.
Exactly. Yeah.
And this is still true, but nowadays we can actually take a whole dendrite skeleton and map onto it the optimal diameters that perform this optimal current transfer from synapse to the root.
So how do you measure this? So some kind of P.
So this is not a measure. This is in the model, right? So
I understand that, but you need some kind of metric for what does it mean to have the same weight? Is it like the amplitude of the APSP, the potential that reaches the soma, or
you could use synaptic inputs. What really works very well is just injecting a current, the same current all over, and then making sure that the result as much as possible the same. It can never be completely the same, of course, because distal synapses are going to be less heard. But indeed, with this, it's not super intuitive. But with this tapering, you get effects that help the distal currents to propagate to this.
So this is like a DC current, like a constant current.
So the method that we now have developed, we can test anything, and the DC works and this current transfer optimization. So this democratization that really works in many ways. So it actually reproduces the diameter profiles from real cells. It also, so there was this recently. There was a group, Maija Liao and Jonathan Howard in Yale. They showed that the local diameters in dendrites, they have a very strong correlation to the size of the subtree at any location in the tree. So you'll have the local diameter. If it's thin, then the subtree is also small. So there's only maybe one or two branches still. If it's thick, then there's the whole tree still behind it, right? And there's a very strong relation there. And when we take their data, their their skeletons, and we find the optimal diameters for current transfer, then we get the same relationship also. So this very strong relationship between the topology and the diameters.
But I guess your analysis is
for now. By the way, this is not yet completely. This is not published yet. So
okay.
But for now, it looks like this is
okay. But this is all then for assuming passive dendrites. I assume
this is passive, but the method. You can apply anything you want. So we also looked at protein transport, which really, we would predict completely different profiles than we actually see. So actually, in our hands, it looks like it would be very good if the dendrites actually grow towards the tip for protein transport optimization, at least under certain conditions. So you can do anything. You could look at propagation of back propagation of spikes or whatever, and find out what would be the diameter profile that corresponds to this.
No, I'm just sort of curious if you find an agreement between your predictions based on this
Yeah.
synaptic, what they call a democracy rule, and passive dendrites. And if that agrees with experimental observation, it means maybe that it is actually is the passive properties that the active dendritic properties
Right.
doesn't really
Yeah.
affect this aspect so much.
Yeah.
It obviously.
So surprisingly, for our morphological models, we don't have to put any real sophisticated computational constraints. So neurons have a computational function. They do coincidence detection or specific integration of inputs on the dendrites. And I'm sure Poirazi talked about this in the
Mm,
previous podcast. Although I didn't yet have time to read it to hear it. But
mm,
uh, um, our morphological model doesn't require that we put in these constraints.
mm,
So, in my view, and this is after working with these models for a long time, actually, the morphology of dendrites is strongly determined by the connectivity that it implements
mm,
than by the actual function, except for these very basic, like conduction time, right? That's an
uh,
obvious one, and these current transfer. Um, so more
mm,
sophisticated computations, I think, then also come from the fact that, well, you have a spatial distribution anyway, because you have
mm,
to send out your dendrites, and then specific distributions of nonlinear ion channels,
mm,
voltage gated or whatever. They then implement probably the sophisticated computations that
mm.
dendrites do
Yeah.
implement.
Oh, no, but that's good. It sort of is. I think any observation, any principle that seems to be robust and can be understood on rather basic principles, that's good news. Maybe sometimes we sort of get so. I think in the field we get so overwhelmed by these possible non-linearities that we sort of lose faith a little bit. But often things are simpler than you might think. That you can actually understand it on, like say in this case, maybe with passive dendrites and
But I
so.
would be very interested, of course, and excited if I couldn't predict things with just these passive
Yeah,
passive assumptions, because then we would find out something probably interesting.
yeah.
And in certain cases, there's some examples of this.
But
But yeah.
interesting in neuroscience often means some complication. So it's good maybe to get sort of like a
Yeah.
exciting paper out, but maybe not good. It's one more thing that you have to worry about
Yeah.
at the
So
moment.
in general, indeed, like also, for example, these connectome data sets, they're
Mm.
so huge. They give you the whole data for the whole circuit of the whole brain in the fly. It's very complicated. So
Yeah.
it's nice to see, and we want to use our models to make at least a simple prediction of, if our rules are the only
Mm.
thing that matters, this would be the
Yeah.
prediction. And then probably some things won't match, and those will be the interesting things we have to dive into
Yeah, exactly.
further.
And just sort of like these are exceptions to the rule,
Yeah.
but nevertheless. So, but how, I mean, have you when you work on, I don't know, this fruit fly data, right? Which is sort of really
Hmm.
pushing the envelope, I think, in terms of like, I mean, it's like the amount of work going into it and the amount of data. I guess there's like
Incredible.
petabytes or
Yeah.
something of data or something. So, how are you able to? I mean, how do you deal with petabytes of data if you want to do research on them? You have to. I mean, I don't know how. I don't really how it's set up, right? I mean, I think the some of these new initiatives are really good at making good user interfaces. So hopefully
Yes,
that's all the
but
case
you
with
have
the
to
fruit
really
fly.
be very focused on specific parts of the circuitry, and ask
Yeah,
very specific questions. Otherwise,
yeah,
you get overwhelmed. And
yeah,
by the way, of course, also not all the neurons are they're all reconstructed automatically, but you still have to double check
yeah,
for the specific circuit component that you are particularly interested for a
yeah.
particular question,
Mm.
that they are good. So you have to be you
So
have to be careful when you make
yeah,
big assumptions about the
yeah,
whole circuit. Yes. Yeah,
mm.
it's but there's a lot of people working on this, and
Yeah.
it's
Well,
great.
which is great.
Yeah.
Yeah, it's like keep they keep the scientists busy for a long time. Yes,
Exactly.
yeah, we just can extract as much information as we can about yeah, from this beautiful data set. So, and then one thing we haven't talked so much about is, I mean, you talk about growing neurons, but how to grow networks? Then how do
Mhm.
you, yeah,
Right. So as I just mentioned, it will be interesting for us to go from the single neuron that's grown in basically empty space. It's completely artificial, of course,
mm,
to this space-packed system that you have the data from in the connectome, where dendrites also grow around structures that are in the way, and they grow together. And so we are working on this, and I think we're making progress. But there are some obvious difficulties along the way. So first of all, it was important that we now figured out what is the actual developmental process of growth. So
mm,
we've figured that out for a very select few cell types, and we identified kind of a general growth principle that roughly works
mm,
for other cell types as well. But if you want to simulate, let's say, the whole brain, then you'll need to have that for all the cells involved. That's a challenge. But also, we still grow branch by branch, and we don't have a continuous outgrowth. So we're now working on a way that grows more and more like the reality, right?
mm,
So micrometer by micrometer, and if you already just this switch already involves a lot of complications, because now suddenly the cell needs to know global things that it has no way to actually understand about where
yeah,
it will be allowed to grow into. Like how does it identify this innovation area, for example,
yeah,
and how does it really still conserve the optimal wiring while growing bit by bit, and several branches at the same time
yeah,
that need to shouldn't grow along the same way, and these kind of things. So
mm,
once we have that, then there's other algorithms from graph theory that allow you, like from Dijkstra, for example, that allow you to find the best path around obstacles. This we have done and shown that it's possible. So once we have these developmental algorithms, we will be able to use these ways to actually simulate the space packed tissue for simple structures.
mm, yeah. Cool. So, of course, you immediately when you published this 2010 paper, you made this, which I think is very commendable, this trees toolbox, so that people can grow their own dendrites. And then in 2017, you came something called T2N,
That's right.
which is some kind of what is that? Some kind of
So,
merging of neuron and trees.
exactly. So Trees Toolbox is a MATLAB package that's relatively general, and even allows a little bit of this passive
Mm,
electrophysiology stuff that we talked about already today. But the more sophisticated modeling with spiking and nonlinear, dendritic spikes and stuff, they need to be done in the separate simulation environment. There's neuron, and
mm,
so we actually wrote this tool to link the two, where you basically stay in the MATLAB environment and don't see neuron. So you'll send your morphology and everything to neuron, and then you get the results of the simulations. And so what is the computation that the dendrite actually performs? You can do that, and then
mm,
you can, for example, then change the morphology with this changing parameters in the morphological model, and study the structure function relationship.
yeah.
And this allowed us actually to generate, I think, a new kind of compartmental model. So as you said earlier, traditional compartmental models are um, done by taking one morphology that's being drawn from microscopy, and then putting some ion channels and everything, and then studying the computation or the electrophysiology of that particular
Mm.
morphology, right?
Yeah.
And with our T2N, you can go a little bit beyond that, because you can actually generate compartmental models that work on this entire morphological space of possible, for example, granule cells in the dentate gyrus. That's something we did, but we also looked at other pyramidal cells in the CA1 in hippocampus, and also pyramidal cells in the
Mm.
cortex, etc. And so we have a possibility to actually really manipulate the morphology in a functioning compartmental model. That's not so obvious. It sounds maybe simple, right? You just change the morphology and see. But often these compartmental models are written in a way, after micrometer 212, that's when you put this
I see.
particular ion channel for
Yeah.
100 micrometers, and then if
Mm.
you write your compartmental model like this, then it doesn't generalize
Mm.
to any morphologies, right? And so we've had this problem typically, that we manipulate the morphology and then the cell
Mm.
stops, even changing just by a few more micrometers the morphology, the cell stops spiking altogether. So they're not very robust. Whereas then the models that we generate using the T2N, for example, they are robust for many for large space of morphological parameters.
But what
Um,
about the spines? Is that something which is part of
yeah,
your?
not yet.
No.
This um, we have in 2010. I really put everything that I could think of. So
Uh,
you can distribute spines, but they're very artificial, and we haven't
uh,
studied much this relationship between spines
no.
and the morphology of
Yeah.
dendrites. What I should still say is there are now efforts from the neuron developers as well. Robert McDougall and others
Mm.
uh, to make an interface between MATLAB and neuron,
Mm.
and so we are probably going to link this whole thing so that we can use uh, the real inherent neuron functions
Mm.
instead of just our hack, which just sends
Mm.
requests to neuron and collects
I see. So
them.
yeah. So but I mean now, Mrs. I well, the hope is that this podcast reaches computational neuroscientists all over the world, and not everyone has access to MATLAB. Is
That's
like a
right.
Python thing on the way, or?
Yes, I mean we we have started working on that, but we really it's a lot of code, and it's all in MATLAB. And I
uh,
started with MATLAB, and I really like MATLAB as well. I mean,
yeah.
that is there have been some efforts to translate, for example, these minimum spanning tree based
Mm.
algorithms into Python, and they're too slow. Basically, you cannot use them. So
Ah,
you'll have to go deeply into C or some other ways to do this. But I think the future is nice, because I think translating from Python to MATLAB or the other way around is going to become fairly easy in the future with, for example, artificial intelligence kind
yeah.
of
That should
tools and
be.
large language models.
That must be the hope. So then you, yeah.
We are always exploring this
Yeah.
option also, but
Okay.
yeah, yeah.
So one thing, it's a lot of code, but it's also that it's yeah, it's not so easy to make it as efficient in Python without going into like this some more like some kind of low level languages and stuff.
Yeah.
Yeah.
So for now, unfortunately, if you want to take advantage of I don't know how much it's like 200, 300 functions that we have, you have to use MATLAB. But there's also ways to use MATLAB freely in the browser.
Mm.
So that's actually
Is that like Octave you mean, or?
no, it's you can actually use MATLAB from MathWorks
Oh,
online version.
oh, oh, free? Oh,
Yeah, I to a certain degree, right? I mean,
Yeah.
this restricted
Yeah. Okay.
usage,
Something.
but
Oh, I
yeah.
see.
So for using the trees toolbox, I think it's definitely good enough. So developing the trees
Yeah.
toolbox, there's good
Uh, yeah.
good reasons to then have the
Yeah.
real version.
So I mean, now I think this is really really cool. So I think this is a great important subfield in computational neuroscience to develop this thing. So how many people are working on this? I mean, uh, this kind of models.
So it's really mostly it's our lab, right, and then
Yeah.
some collaboration partners. So it could be much more. There are new developments. There's a really nice way to grow trees. That's a new way by Lida Canari from the Blue Brain. She's now in Oxford,
Mhm.
I think, and she uses a completely different topological approach to generate morphologies. But again, yeah, that there's not so much going on
No,
there,
but it's
right?
so interesting because this is
Worldwide,
so.
let's say.
Yeah, I think this is very typical, right? I mean, this is obviously an important field of a subfield of computational neuroscience, and still there are very few people working on it
That's right.
because computational neuroscience is a sparse field. People are sort of working on, at least there's not enough people to properly work on the problems that should be worked on. I think. Uh,
Well, I mean, of course, other problems are also important, right?
absolutely, but so it's not.
But it is true that I think it's kind of this. Uh, now you can say traditionally, I think electrophysiology and the function have been in people's minds, right?
Hmm,
Whereas anatomy has kind of this kind of boring flavor, and this is changing dramatically. But in the modeling world, it hasn't completely arrived yet, I would
yeah,
say.
yeah. No, I think that's true. So so I'm not saying that other people in computational neuroscience who don't work on this are doing the wrong thing. It's just that we could have much more people in the field as a whole,
As a whole, absolutely,
and
absolutely.
then we can do all kinds of things. So that
But
is
even
cool.
that is getting easier, right? Because programming is has become easier to learn. We have a lot of new people who come with very little programming experience, and
That's
within a
true.
few
But
weeks they can
of
do.
course, there is sort of like this new exciting field of using AI in neuroscience, neuro AI. So
Yes.
that is sort of like that's another thing that we spread our resources on. Of course, I guess it's also lots of resources coming in because it's AI. So I don't know, but
Yeah,
anyway, so
yeah.
it's
We'll have to see how that develops and
yeah,
whether how
that's true.
much it's worth it. Yeah.
So I think, I mean, if you sort of look back at Hodgkin and Huxley, I mean, obviously one thing they put into their model was sort of like these ion channels, sodium and potassium, and for the opening and closing of these gating variables, they used sort of statistical models, and now an alternative approach is sort of to use molecular dynamics to try to understand essentially the opening and closing of these models, even though it hasn't. I don't think it's very difficult to do because the long time. I mean, if you want to simulate
Yeah.
these ion channels on a molecular scale for milliseconds, then it's it really you don't really have computers for it. But anyway, the goal must be to one goal is sort of to try to also make mechanistic models and connect these statistical models to
Mm.
what's going on the lower or more mechanistic models.
That's
Uh,
that's of course important and definitely more possible now with better computers. But you have to also be careful that you don't lose yourself in the details. So obviously,
yeah.
also for dendrites. So we model dendrites as basically in compartmental models as connected pieces of
Mhm.
cylinders, right? And cylinders, cables that have a membrane where the current flows through the membrane and then or can flow inside, and their cylinders are circular, right? Have a
Mm.
circular diameter, and you could also simulate the ion movement inside the mesh of the realistic mesh,
Mm.
but you have to be careful that you don't uh, you don't make your simulations too complicated or more complicated than necessary. Depending on the questions you ask,
That's true,
this is going to be completely irrelevant, right?
for
These
sure.
small
But that,
details.
but that's sort of like, I would say, like these are sort of two mechanistic models at different levels of coarse graining, right? And you're
Yeah.
saying that we don't need all this. We can deal with the simple model of the cable. But I'm sort of
Exactly,
my
but in a spine, for example, the very small structure,
uh,
there you really have to be very careful how the exact shape and the mesh that describes the
sure.
shape of the spine
Yeah.
determines the
Mm.
distribution of calcium and other things.
And also there the number of calcium ions inside the spine is often so small that you
Yeah,
it's sort of assume
surprisingly
modeling as a
small.
concentration. It's sort of
That's right.
rather than just keeping track of the three ions in there, whatever. Yeah. So
Yes.
that's. But I'm also more I'm sort of well, I was sort of leading up to a question sort of now. What you talked about so far is statistical models
Hmm.
of dendritic growth, or at least not mechanistic models.
Mm.
And but how close are we to mechanistic models of dendrite growth? Do you follow that field? I mean, like with growth cones and molecular signals
Yeah,
and
not so. That is for us still quite far off to
uh,
study. The particular aspects of growth cone splitting or something like that by
uh,
molecular processes or so. Or I mean, but we have on occasion people who think, yeah, okay, maybe there's molecular gradients on which
yeah.
branches may travel
Mm,
in particular ways, and that could describe the growth process in some ways, or make it more mechanistic in your terms. But yeah, it's I think there's still something fundamental to crack. So um, we have this one very strong scaling relationship that we observe in our model that's determined by optimal wiring, right? There's a this two-third power between the length of the dendrite and the number of branch points, or the number
okay.
of carrier points,
Mm,
and there's a particular relationship also with the volume that the dendrite innervates. And we have tried in several projects to generate this kind of random growth that follows maybe molecular gradients or self avoidance, things that are more
mechanistic.
close to biology.
Yeah.
Um, and we were not able to find any combination of parameters that actually
Yeah.
spits out this two-third power, this particular scaling relationship,
Mm.
except if you actually put it in. Of course, if you put it
Yeah.
in, then that's possible, right?
It
But
doesn't go. Yeah.
that's
Mm.
not very satisfying, right?
No.
So there's some fundamental
Yeah.
missing link there between the
Mm.
two things, even at
Yeah.
this level, without considering molecules or anything. It's
Yeah.
even a
Mm.
random growth. We were not able to do this. So yes, I think from biophysics. People following microtubules and actin and
Mm.
there's certainly more and more that will be feasible and
Yeah.
and uh,
Sure.
but that but we're not there where we can actually make that
Big. Yeah.
jump
Oh.
quite yet, except if you want um, the kinds of things we do with Gaia Tavozanis, right? She can actually make some mutations on particular aspects, for example, of actin regulatory
Oh.
proteins, and then see how that affects the morphology, and there we can see what maybe link aspects of our model
Mm. Yeah.
to molecular.
Mm.
but this is still in the infancy, I would say.
Well, there's plenty of work to be done and plenty of exciting discoveries to be made. So,
Yes.
but back to your, your sort of most like your ongoing projects. What I guess you're still working on aspects related to this. What's on the horizon in your lab?
Yeah, I mean, a lot of the things I've mentioned now are still ongoing, actually. So
That's good.
the big question, uh, indeed, or the big technological project is to get these fake connectomes going, basically,
Mm,
and we're putting going several parallel ways to get there, and then. And then we have, of course, through our uh, tight work together with Peter Jedlička, we have this a lot of projects that link the morphology
mm,
and the compartmental models, the predictions, what aspects of dendrite shape actually determine the computation, and there's some really very funny, simple things that we found that were that came out from this work together. So we published recently this dendritic constancy idea, which is which goes a little bit against the idea that dendrite shape determines the computation that the dendrite
mm.
performs. um, uh, it What we found is if you keep everything the same, like the average diameter of the cell, the synapse density, um, and the way the synapse strength and the way that the synapses are being activated. If you now activate many synapses in the very same way in and use completely different morphologies, very small ones, very big ones, very branched ones, less branched ones, etc., then actually the input output function stays pretty much the same. So
Ah,
and for us this was surprising, not necessarily for for example, for Idan Segev, who was a cable theory expert. This it was inherently people knew or had a feeling, intuition, let's say,
ah,
that this is true. But so we were able then to using the morphological model to actually explore this in
cool.
detail. And now of course this opens up all kinds of questions. So we actually saw that in Alzheimer's model, in a collaboration with Stefan Remy, we saw also, okay, the smaller cell actually in our model prediction, it should actually behave the same way because of dendritic constancy. But it doesn't. So in the
Mm,
Alzheimer's model, the pyramidal cells in the hippocampus
ah,
are smaller, and so that then must mean that there's other aspects that play a role than the change in the morphology.
So, I mean, but dendritic constancy, if you think of how that can be useful for the brain, it sort of is that it's like it's sort of like a synaptic democracy kind of thing. I-
Um,
Is that it? So it's can you sort of think of like
so,
that it's something which is useful, and if that sort of is breaks down in Alzheimer, then
right.
that can explain some of the I don't know, maybe that pushing it too far.
So it's a baseline. It's convenient. Let's say we have a cell that grows during development, right? And
Mm,
there's much more dendrite cable, more synapses
yeah.
as well. You don't want the function of the cell to change, right?
Oh,
You want that to stay the same. And
yeah.
so if the way that it grows keeps the density of synapses the same, and then all the other properties the same,
Mm,
then
yeah.
rigid constancy says the input output function in terms of numbers of spikes is going to be the same. Um,
Hmm, cool.
Other things like temporal patterns of spiking, they would be affected. Some
Okay.
people, of course, argue that the timing matters less, and maybe
Yeah.
firing rate more. Probably some cells,
Mm.
temporal aspects matter more, etc. But in any case, the firing rate as a response to these distributed inputs would stay the same. Yeah. And that actually would be also the case in the disease case. So if you have a lesion or if you have Alzheimer's, it would actually keep the level of activity, make sure we don't get into epilepsy or so, because otherwise, like, in principle, the um, yeah. So what was the consensus before, let's say, the dendritic constancy, and of course it still is, but is if you inject just one current and you don't consider synaptic inputs, you inject one current into the soma, right? You have a small cell that has little membrane, so you push a current in. It's hard to push the current in. The voltage gets very high very quickly. And if you have a large cell, you inject the same current, it flows through the membrane easily, therefore you don't reach the voltage to spike. So the smaller cell is, in terms of current injection in the soma, is the smaller cell is much more excitable, right? But in our dendritic constancy context, if you have synaptic inputs, then actually this additional leak through having more membrane is compensated for by having more synapses, so more current, and these two things, fall out of the equation, and so then the input output function stays the same. So that's one thing that we kind of found, if you want, with combining these
But
models.
one question. I mean, if you you look at dendrites. I mean, like when I talked to with like Jota Barazzi in the previous podcast. I mean, you have these active conductances and these regenerative processes, like a sodium spike, and you have a calcium spike, and also an NMDA spike.
Right.
So this is not really what you're talking about here. It's more like the sub-threshold activity.
So all these other aspects, these nonlinearities in the dendrites, but also, for example, gradients in the synaptic weight or actual synaptic plasticity that will strengthen a particular subpopulation of synapses. They, of course, affect the input-output function dramatically. But as a baseline, if everything is the same, then and you have none of those crazy dendritic channels.
But maybe these are sort of like what we mentioned. These three sort of regenerative. That's like supra-threshold activities, right? So maybe for the sub-threshold, even though there are sub-threshold dendritic conductances, maybe they don't change this aspect so much. That's sort of like I'm just rooting for simple principles there, so that we can think of it.
Well, what actually can be said about this is that we took Yota's model, hippocampus here, one pyramidal cell, and we translated it to T2N, to our pipeline, and then changed the morphology, and so then use different pyramidal cell sizes, and so the spiking, even in this very model with complicated dendritic spikes, so the number of spikes were unchanged if you look at different sizes of cells. And even the dendritic spikes, the number of dendritic spikes were also conserved. So dendritic constancy, but that's incidental. I mean, this is just this one example.
N equals 1. Yeah.
So there is ways even to preserve, I guess, these dendritic nonlinearities, and this remains to be seen how potentially some of those aspects, like dendritic spikes and NMDA spikes, and also gradients of synapses, whether they actually maybe even contribute to constancy or democracy. So actually keep things more equal than making them more complicated. Who knows?
So this has been very exciting, and I think it's the very important work. So I'm happy that people now get to know more about it. Before we end here, do you have anything we should add? Anything that you're
No,
no?
thank you very much. It was very pleasant and very nice.
Okay. So then, well, thanks a lot.
Thank you.